This is an article by Peat called Aging Eyes, Infant Eyes, and Excitable Tissues. This is on his website, raypeat.com, and you can read it there yourself. This is how it goes. The eyes and the lungs are sensitive tissues that are easily harmed by inappropriate environmental exposure. They are especially sensitive in infancy and old age. For 60 years, there have been controversies about the cause of retinopathy and prematurity, which has blinded tens of thousands of people. Degeneration of the retina is the main cause of blindness in old people. Retinal injury is caused by ordinary light when the eyes are sensitized by melatonin, prolactin, and polyunsaturated fats. Bright light isn't harmful to the retina, even when it is continuous, if the retina isn't sensitized. Melatonin and prolactin are induced by stress, and darkness is a stress because it impairs mitochondrial energy production. The polyunsaturated fats which accumulate in the brain and retina damage mitochondria. Iron, which accumulates prenatally and then again with aging, reacts with unsaturated fats during stress to destroy cells. The popular supplements melatonin, tryptophan, fish oils, St. John's wort, and the various omega-3 oils all increase the risk of retinal light damage and macular degeneration. Serotonin uptake inhibiting antidepressants are suspected to be able to cause it. Processes similar to those that damage the oversensitized retina can occur in other cells as a result of stress. The substances that sensitize the retina to light damage can also increase the incidence of new or metastatic cancers. Iron supplements and the use of supplemental oxygen, especially with a vitamin E deficiency, exacerbated by excessive unsaturated fats in the diet, are still commonly used exactly when they can do the most damage. One of the recognized achievements of biology has been the demonstration of life's universality. I think I said that wrong. Universality. In the sense that organisms of all sorts use the same fundamental genetic code, and that yeasts, lizards, apes, and people have remarkably similar cellular systems, as well as a great amount of genetic similarity. There has been another, less well-recognized sort of convergence going on in physiology and pathophysiology. Hans Selye's concept of stress, the syndrome of being sick, Otto Warburg's argument that a respiratory defect was behind all kinds of cancer, and the idea of free radical damage as a common factor in disease and aging, helped to create a more general way of looking at the nature of disease that superseded medicine's theories of disease pathogens and genetic mutations, which created thousands of disease entities, none of which had much to do with the individuality of the patient or his environment. The understanding that plants and animals have much biochemistry in common has gradually changed the assumptions of the science establishment, which until recently insisted that only ionizing radiation could affect animals or other organisms that lacked chlorophyll, and insisted that ionizing rays acted only on the DNA. Visible light, the textbooks said, was not chemically active, and so couldn't possibly affect animals' cells. In animals, coloration was seen mainly as decoration and disguise, rather than as a functional part of their biochemistry. Chemically, the meaning of a pigment is that it's a chemical which selectively absorbs radiation. Old observations such as Warburg's, that visible light can restore the activity of the respiratory pigments, showed without doubt that visible light is biochemically active. By the 1960s, several studies had been published showing the inhibition of respiratory enzymes by blue light and their activation by red light. The problem to be explained is why the science culture simply couldn't accept crucial facts of that sort. The retina, of course, was allowed, in the views of mainline science, to respond to ordinary light, but the few people who studied the biological effects of seasonal or daily cycles of light have until recently stayed very close to the nerve pathways leading from the retina to the pineal gland, because those pathways could be described in terms of an evolutionarily specialized third eye. Even with a doctrine of a genetically specialized link between the retina and a little of the animal's physiological chemistry, the great, slow-witted science establishment has done its best to avoid thoughts of any deep interaction between an organism and its environment, by insisting that the organism runs according to a genetically determined clock, which is located in a few cells in a certain area of the brain, and that nervous impulses from the retina have only the small privilege of, quote-unquote, setting the clock. It didn't matter to the academic and medical worlds that a professor, Frank A. Brown, had long ago disproved the idea of an innate genetic clock, because philosophy is much stronger than evidence. And I'm not going to say this right, but Leibniz, I believe is how you might pronounce it, Leibniz had said that everything in the world runs on its own inner clock without needing to perceive its surroundings. And this idea that everything in the world is a windowless monad resonated through the world of science, because it justified the pompous authoritarian attitudes of the experts who knew that anything that wasn't already in their heads couldn't be considered knowledge. If an organism's essence is contained in its genes, then it clearly doesn't interact in any meaningful way with most of its environment. This is the sort of culture that imbued research on the biology of light cycles. When I moved from Mexico, first to Montana and then to Oregon in 1966, I became very conscious of how light affects the hormones and the health. For example, in Montana I experienced an interesting springtime shedding of body hair. Many people who came to cloudy Eugene to study, and who often lived in cheap basement apartments, would develop chronic health problems within a few months. Women who had been healthy when they arrived would often develop premenstrual syndrome, or arthritis, or colitis during their first winter in Eugene. The absence of bright light would create a progesterone deficiency and would leave estrogen and prolactin unopposed. Beginning in 1966, I started calling the syndrome winter sickness. But over the next few years, because of the prominence of the premenstrual syndrome and fertility problems in these seasonally exacerbated disorders, I began calling it the pathology of estrogen dominance. In the endocrinology classes I taught at the National College of Naturopathic Medicine, I emphasized the importance of light and suggested that medicine could be reorganized around these estrogen-related processes. If the sparrows of Times Square made it in the winter because of the bright lights, it seemed clear that the bright artificial light would be helpful in regulating human hormones. In our lab at the University of Oregon, our hamsters would try to hibernate, even though they were in temperature-controlled laboratories with regular cycles of artificial light. The ceiling lights provided only dim illumination inside their cage boxes, so they were probably in a chronic state of light deprivation, which probably increased their sensitivity to the weak environmental cues that Frank Brown had investigated, possibly microwaves that easily penetrated the lab walls. During the winter, when they were infertile, I found that their thymus glands practically disappeared. The mechanism seemed to include the increase of pineal gland activity, probably increasing melatonin synthesis in the winter, under the intensified activity of the sympathetic nervous system, with increased activity of adrenaline and other catecholamines, and the melatonin was apparently a signal for suppressing fertility during the stressful winter. In some animals, and this is from a study by, I'll say these incorrectly, but I'll try, Shavarva and Navratilova in 1989. So in some animals, estrogen is increased during hibernation, contributing to the reduction of body temperature. In 1994, A.V. Sirotkin, Sirotkin, found that melatonin inhibits progesterone production but stimulates estrogen production. and it's widely recognized that melatonin generally inhibits the thyroid hormones, creating an environment in which fertilization, implantation, and development of the embryo are not possible. This combination of high estrogen with low progesterone and low thyroid decreases the resistance of the organism, predisposing it to seizures and excitotoxic damage, and causing the thymus gland to atrophy. Cyclical exposure to melatonin can have an effect on the reproductive system opposite to that of chronic exposure. And the way exogenous melatonin is delivered to the animal can have unexpected effects on the actual amount of melatonin circulating in the blood. This was from Wright and Alves in 2001. The actual amount of melatonin in the tissues, its relation to the normal cycling of the animal, and the influence of temperature are often disregarded in melatonin research, making it hard to interpret many of the publications. There's a lot of talk about melatonin's function as an antioxidant. But like so many other antioxidants, melatonin can act as a pro-oxidant at physiologically relevant concentrations. Some studies have found that it, like estrogen, increases the activity of the pro-oxidative free radical nitric oxide, which acts like melatonin on pigment cells, causing them to lighten. The promoters of estrogen are also making claims that estrogen is a protective antioxidant, though that isn't true of physiological concentrations of estrogen, which can catalyze intense oxidations. The market cultures seem to guide most research in these substances. Almost any kind of stress increases the formation of melatonin. In some animals, melatonin has been shown to be responsible for whitening of the hair during the winter. In some species, it acts directly on the pigment cells, but in other species, it seems to inhibit the action of the melanocyte-stimulating hormone. In snowy climates, it's ecologically rational for animals to turn white in the winter for camouflage. But tadpoles also turn white in the dark or under the influence of melatonin, and the biological meaning of that isn't so clear. It's possible that being white would reduce their loss of heat through radiation, but I think it is more likely that it relates to an increased ability of weak radiation to penetrate their tissues, rather than being stopped near the surface by the melanin in the skin. The absence of melanin makes them more sensitive to light. Bright light suppresses their melatonin and makes them turn dark brown or black, and this protects them from bright sunlight. In the retina, melatonin increases the sensitivity of the cells to dim light. It, along with prolactin, another nocturnal hormone, helps to produce dark adaptation of the eyes. Melatonin increases the concentration of free fatty acids during the night. This is from John et al., 1983, John and George, 1976. So it's interesting that one of the long-chain, highly unsaturated fatty acids, DHA, or docosahexaenoic acid, also increases the light sensitivity of the retina. Melatonin lowers body temperature, causes vasoconstriction in the brain, heart, and other organs, and slows reactions. An antagonist to melatonin acts as an antidepressant, reducing behavioral despair resulting from stress. This is from a study by Dubosevich et al. From 1990. So in the behavioral sense, melatonin reduces sensitivity, yet it increases the eye's sensitivity to light, causing them to be injured by light that would otherwise be harmless. Since a hibernating animal under the influence of melatonin can become very cold, the light sensitizing function of melatonin is probably related to the biological need to be roused out of the torpor occasionally. Hibernators apparently have to warm up occasionally to sleep in the ordinary manner. Melatonin is said to intensify dreaming, which is part of the process of arousal from sleep. All of the stress-related hormones increase during the night. One of the ways these hormones of darkness act is to increase the sensitivity to light, in a process that is an important adaptation for organisms in dim light. In the night, our ability to see and respond to dim light is increased, but dark adapted eyes are very sensitive to injury by bright light. Light that ordinarily wouldn't harm the eyes will do serious damage when the eyes are dark adapted. In thinking about the effects of stress and oxygen deprivation, I read the studies demonstrating that the formation of the oxygen-wasting age pigment, lipofuscin, is increased by estrogen, by oxygen deprivation, in carp living below the ice or even in fetuses, by metals such as iron, by X-rays, and by highly unsaturated fats. Free fatty acids that are mobilized from stored tissues in the night and in the winter also tend to increase with aging, as the ability to tolerate stress decreases. Poor circulation and lipofuscin tend to be associated in a vicious cycle. This means that the retina becomes easier to injure by light in old age, for some of the same reasons that the infant's retina is susceptible. The fetus accumulates a very large amount of iron, and it absorbs melatonin from the maternal circulation. Prolactin is sometimes elevated in the newborn. Premature babies are often given extra oxygen, which tends to cause vasoconstriction by displacing carbon dioxide. Melatonin's ability to cause vasoconstriction means that stress makes supplemental oxygen more toxic. Synthetic glucocorticoids are often given to premature babies, adding to the risk of retinal damage. When the mother has been given iron supplements during pregnancy, along with unsaturated oils in the diet, the baby is likely to be born with a vitamin E deficiency and suppressed thyroid function, increasing the probability that it will be jaundiced, leading to treatment of the jaundice with exposure to very bright light. Although Yandell Henderson had already, in 1928, explained the need for carbon dioxide to be used with oxygen for resuscitating infants or adults, medical researchers and hospital workers could never accept the idea, probably because of a fundamental misunderstanding of the Henderson-Hasselbalch equation. Animal experiments show that supplemental oxygen with carbon dioxide causes vasoconstriction, reducing the tissue's supply of glucose as well as oxygen. In combination with too much light, especially blue light, it damages the retina. At hyperbaric pressure, oxygen causes seizures as well as damage to the lungs and other tissues. The contribution of bright light to retinal damage in babies has been denied in several recent publications, and these articles undoubtedly provide useful material for defense lawyers to use when hospitals are sued for causing blindness. One publication based on experiments with kittens concludes that bright light does not harm the newborn's retina. But the comparison is between continuous light and intermittent light, rather than between bright light and dim light. 12 hours of total darkness, rather than sparing the eye by reducing its exposure to light, would sensitize the eye. The only reason such appalling things can be published is that their conclusions protect the hospitals. A few good studies of the effect of bright light on the retina and the fact that dark-skinned people with more protective pigment in their eyes have a lower incidence of retinopathy and prematurity make it clear that the ordinary laws of physics and chemistry actually do apply to the infant eye. Light and stress, especially with excess iron, damage the retina when the cells contain too much PUFA, since these fats react with light and free radicals. The nocturnal stress hormones, especially prolactin and melatonin, make the retina more sensitive to light and more easily damaged. It's too much darkness that sets up the problem, since the eyes will adapt to excess light, but darkness increases their sensitivity. The use of lasers to operate on eyes produces intense inflammation of the eye, but even at low dose, the diffusing light causes retinal macular damage. Cytochrome oxidase is one of the enzymes damaged by stress and by blue light, and activated or restored by red light, thyroid, and progesterone. It's a copper enzyme, so it's likely to be damaged by excess iron. It is most active when it is associated with a mitochondrial lipid, cardiolipin, that contains saturated palmitic acid. The substitution of polyunsaturated fats lowers its activity. Mitochondrial, mitochondrial function in general is poisoned by the unsaturated fats, especially arachidonic acid and DHA. Creating a deficiency of DHA, even when an oil of known toxicity is used to replace omega-3 oils, prevents retinal damage from light. Despite evidence of this sort, Mead Johnson is going ahead with the marketing of its baby formula containing added DHA, which is industrially extracted from algae. Although the researchers who claim that DHA is beneficial haven't answered my letters, a representative of the company that manufactures it did answer my question about the actual composition of the oil and acknowledged that they don't have any idea what the minor ingredients might be. When animals are made quote-unquote deficient in all the exogenous polyunsaturated fatty acids, linoleic and arachidonic acid, as well as linolenic and DHA, they become remarkably resistant to all sorts of stress and toxins. The polyunsaturated fats make the lungs more sensitive to excess oxygen or hyperventilation. They make the eyes more sensitive to light, and they make the brain more sensitive to fatigue. The use of synthetic glucocorticoid hormone is standard in treating very premature babies, although it is known to contribute to eye damage. This is because it is considered necessary to improve the lung function of premature babies with respiratory distress. But there is no clear evidence that it is beneficial for lung function in the long run, and very clear evidence that it damages the brain and other organs. There is widespread agreement regarding the use of the glucocorticoids prenatally to accelerate lung development in women who seem likely to deliver prematurely. Natural cortisol is a factor that promotes lung development prenatally, but cortisol is also a signal produced by a stressed fetus. That triggers the birth process. Cortisol, or the synthetic glucocorticoid, inhibits progesterone production and stimulates estrogen production, activating uterine contractions and other processes that terminate the pregnancy. Apparently, it doesn't occur to many people that administering the glucocorticoid triggers premature birth, creating the problem they are intending to treat. Recognizing causal connections between premature birth and respiratory distress and retinopathy of prematurity, it would be obvious that the greatest efforts should be made to prevent the problems by improving the health of pregnant women. Hospitals, however, are invested in high-technology symptoms for treating these problems, and even though the results are dismal, they can't make money by getting pregnant women to eat enough protein to prevent preeclampsia, which is a major cause of premature birth, or by treating the problems with salt, magnesium, progesterone, thyroid, and aspirin when the women haven't had a good diet. Historically, preeclampsia has been blamed on the mother's or fetus's bad genes, and that cultural bias was the setting in which these high-technology prenatal and neonatal systems developed. High-technology neonatology derives from the same ideology that motivated Joseph Mengel's genetic research in Auschwitz. The idea of genetic determination is still motivating resistance to reasonable preventative approaches. Thyroid, i.e. T3, is very effective in accelerating lung development in the fetus, and it doesn't have any of the harmful effects of the synthetic glucocorticoids. It normalizes the hormones, increasing progesterone and decreasing estrogen, which are needed for full-term gestation. The opposite of the glucocorticoids effects. While the cortisol-like drugs damage the brain and other organs, thyroid and progesterone protect them. Old organisms, like newborns, are easily injured by all sorts of inappropriate excitation. As in premature babies, the aged eyes, lungs, and brain are especially sensitive to damage by stress. But all organs are subject to the same kind of damage. Medical treatments for respiratory distress and macular degeneration in old people are often the same as those used, so inappropriately, for babies. The good health practices that can prevent the inflammatory and degenerative diseases can often make it possible for damaged tissues to recover, even in old age. The pituitary hormones, especially prolactin and TSH, are pro-inflammatory. The darkness increases TSH along with prolactin. So to compensate for a light deficiency, the pituitary should be well suppressed by adequate thyroid. Armour thyroid, or Thyrolar, or Sinoplus Cytomel would probably be helpful. Eye drops containing T3 might be a way to restore metabolic activity more quickly. Limiting water intake or using salt generously helps to inhibit prolactin secretion. The saturated fats protect against the body's stored PUFA, and keeping the blood sugar up keeps the stored fats from being mobilized. Aspirin, or indomethacin, is generally protective to the retina. Analogously to its protection against sunburn. Adequate vitamin E is extremely important. There are several prescription drugs that protect against serotonin excess, but thyroid and gelatin, or glycine, as in magnesium glycinate, are protective against the serotonin and melatonin toxicities. Copper and magnesium deficiencies predispose to retinal damage. Red light is protective. Blue light, or UV, is harmful, so wearing orange lenses would be helpful. Progesterone and pregnenolone, by reducing the stress reactions, should be helpful in the eye diseases of infancy and old age, as they are in the respiratory distress syndromes. And then below we have all the references, which is essentially half of this entire article. And I'm not going to read through all of these, as I think Ray's interpretation of them, in my opinion, is quite clear and helpful. This is just for educational purposes only, and I hope that you get some benefit from this audio version of Ray Peat's article, Aging Eyes, Infant Eyes, and Excitable Tissues.